Hello, everyone. Thank you for attending 17 Minutes of Science today. Today I have Dr. Dan Starr from UC Davis. He's joining us. Thank you, Dan, for joining us today. A little bit of background on Dan. Together with one of his collaborators, Grant Luxton, he was recently awarded a Allen Distinguished Investigator Award. And they're working on the LINC complex. They're trying to understand how the nucleus skeleton and cytoskeleton interact in the cell's cytoplasmic matrix. Dysregulation of this interaction is involved in many diseases, including cancer, heart disease, muscular dystrophy, and neurodegenerative disorders. Dan will be using multiple model systems in this grant to gain a better understanding of C. elegans transgenics. Today, Dan will be taking us on a journey where he was before the Allen Award, and where he sees the new collaboration with Gant moving towards this science forward in his lab. Okay, hi Dan, nice to meet you.
Good to see you.
So today in this presentation, we want to sort of ask you a few questions. And one of the questions I want to ask you is, what are the attributes and capabilities does the Allen collaboration bring for you in your lab?
Yeah, so the Allen collaboration is really exciting because it's going to allow us to go in completely new directions. When the instructions for the grant proposal were basically to write a bunch of ideas that would never be funded by the NIH because they're just not conservative. So we took that very seriously. We wrote a bunch of crazy ideas to do a bunch of interesting screens and really to take our labs, our two groups into one, combine them into one group and go a new direction. So I'm trained as a classical C. elegans developmental geneticist. I've been working with C. elegans for over 20 years as a model system, and I always think about questions in cell biology in the context of a developing organism. Gant is more of a biophysicist, where he thinks about individual molecules and he thinks about the mechanics of the cell and the properties inside the cell, though we're both focusing on this complex of the nuclear envelope, the LINC complex, and how it interacts with the cytoskeleton. But since he's coming at it from biochemistry and I'm coming at it from developmental genetics, we can combine the forces into one lab and move forward to try to ask biophysical questions, both in mammalian tissue culture cell systems and induced pluripotent stem cells, and in the C. elegans in the context of a developing organism.
All right. Well, that you answered sort of one of my next questions was what kind of model systems are you going to be using? So that's when you speak about the iPSC cell systems, maybe you can mention a little bit more about how you can use that towards addressing perhaps defects, or how the LINC system works.
Yeah, so Gant has perfected a model system where a nucleus moves in a polarizing fibroblast, and presumably in any polarizing epithelial cell, the nucleus will move backwards, and we can take a molecular approach to what's going on in that and how the LINC complex is then formed. The Allen Institute has developed all these human-induced pluripotent stem cell lines so that you can then induce them to form one type of tissue or another, and you can watch organelles or LINC complexes and their roles at different stages of cellular development. And so they're trying to use these, they're trying to develop this induced pluripotent stem cell line really as a model so you can watch tissue development or cellular development as it changes cell types.
That's nice. And so when you mentioned C. elegans, and how do you see that sort of fits in the complementarity of the data that you'll be generating out of iPSC?
So for me, it's really important that there's limitations in tissue culture cells. These are growing on a flat culture under extreme mechanical forces to sort of spread out in almost a 2D environment. Whereas in an organism, most cells are in a 3D environment and behave very differently than they do on a tissue culture plate, on a stiff stratum. In the worms, everything has got its neighboring tissues, and we find many differences in behaviors of the nuclear envelope and stuff just because they're in a three-dimensional tissue and throughout development. For instance, there's one time in development where the nucleus in the worm, where the nucleus has to migrate from the side of the worm to the stomach of the worm. In order to migrate here, it's got to squeeze through a space that's about 5% of the width of the cell, of the nucleus. So the nucleus has to squeeze through the space and then come out the other side. And we like to think of that as a model of normal part of development, but as a model for how like a metastatic cancer cell might be migrating through the extracellular matrix.
Oh, wow. Okay, so that does make sense for why it's good to look at multiple model systems. In this Allen grant, you've come from your lab tell us a little bit about how the impact that funding did for you in your lab. What was life like before the Allen grant, and what is life like going in the future, and how are you going to commence the grant? So what was life like before?
Oh, we'll ignore the pandemic effects of this question. But before, I've been a one R01 NIH lab sort of for the last 20 years, close to 20 years now, which means I'm usually running between 3 and 6 full-time people, mostly graduate students, but occasionally some really good postdocs, and then a lot of undergraduates and graduate students. And the NIH grants, you sort of got to do what you say you're going to do in the grants in a little bit conservative manner so you can get the next grants, because you're always thinking, how am I going to renew in 4 years the next grants? And the Allen grants allows us to just go do whatever we want to do. And so we're developing forward genetic screens using CRISPR in these tissue culture cells, because I'm a geneticist and I like to think about forward genetic screens. But in this way, we've now tripled the size of our group, and we bring in grants, and we combine our 2 labs, and we'll be, by the end of the summer, up to maybe 14 or 15 people, including 4 new postdocs and a couple new graduate students. And now managing this group is going to be much more different, because now everybody's going to be going off in a different direction, which is going to be a challenge and a lot of fun.
All right, it does sound like a lot of fun. And in fact, tripling the size of your lab sounds like quite a challenge. I mean, how do you attract that talent? Is it difficult to get that kind of talent in the lab? We're talking about
attracting talent. Yeah, attracting talent in the middle of a pandemic turns out to not be easy. But we have one really good postdoc that I'm really excited that's signed to come in the summer, and we have 3 others that are like hopefully this close to signing. And if we get those 4, we will have a team at the end of the summer with 4 young postdocs that are just raring to go and with full of tons of cool ideas that are very well trained from other cell biology labs. And it would just, Gans, I would be ecstatic to get that group.
Oh, that sounds really, really exciting. And so when you imagine where you're going to go with this, what kind of broad impact do you think you're going to see in terms of sort of disease biology here? What kind of areas do you think you're going to have a strong impact if you can sort of meet your aims?
Mm-hmm. So I think at the interface of 2 fields, biophysics and developmental genetics, whenever you're working at an interface, you can find new things real fast. And what we're really looking at is, what are the mechanical properties inside the cell? How are all the organelles sort of interacting with each other inside the cell, and then sort of staying in place, so it's not just a bowl of soup, right? So you can either have a cell that's got a matrix of some sort, and it's sort of like Jello, where everything's sort of in about the right place, things are moving around in directional manners, or you could get rid of that whole matrix and you become liquefied, which is a term the biophysicists don't like to use, but I find the developmental geneticists can understand. And now all of a sudden everything is soup and the organelles are all flopping around. And how does that affect the development of the tissue? Because without a doubt, this is going to affect the behavior of cells in disease. And the mechanical properties are important for regulating all sorts of things, including, even, setting up spindles and setting up ribosomes. Setting up organelles really require on mechanical properties between other organelles and the rest of the cell.
Oh, right on. Well, so it sounds, from the biophysical aspect that you're working on, and you're geneticist. So what is that sort of merger of the tools, the instrumentation, the genetics or the instruments that you'll be needing? And what do you wish was possible, or plan to bring in? What kind of instrumentations are you going to bring in with this?
That's a great question. So we would like to use transgenic approaches, such as things that in vivo biosciences are doing, Biosystems does. And in fact, we've been working very closely with you for the last couple of years to move some of our products forward during this pandemic, and when we can't hire fast enough. It's been great working with you. And we can then take disease associated mutants make the transgenics in our worm system, but then we could also go make it in the tissue culture system and see how that affects the mechanical properties of the cell. And so we want to take it from the tissue, from the system, the whole organism, to the individual cell. And then the common theme in both these places, besides transgenics, is microscopy. So we're doing a lot of cutting edge microscopy, working very closely with our world leading microscope core facility here at UC Davis, where we have a brand new ARIScan microscope that we're doing a lot of live imaging, high resolution. Hopefully we'll be trying to get the new Zeiss laser scanner. That's kind of our dream right now. And so to do a lot of live imaging at super resolution on transgenic organisms would be the approach, I guess.
Oh, right on. Well, we have about another 5 more minutes left in our 17 minutes of science here. Time does go by pretty quick. So, perhaps we've talked a little bit about the Allen Award, and just try maybe a little bit of advice to others, perhaps on this kind of a funding. Where else can some other people kind of approach this? How did you actually get on the Allen Award?
Right. So
can you apply for this? I mean, how does someone get one?
Yeah, so the Allen Institute decided consciously to move into a field of how the nucleus interacts with the rest of the cell, which was not, they then asked about 30 or 40 people if they were interested in applying, including both Gant and I. And I think if I'd written my own grants, or if Gant Luxton had written his own grants, I don't think either one of us would have gotten funded, because we would have just sort of done one off of what we're doing now. But the fact that we came together and we wrote a grant at the interface of 2 fields is, I think, what made this grant so exciting to the Institute, and that they wanted to give us all this money to move forward in the next few years. So my advice is to find a collaborator that pushes you outside of your comfort zone, and that the collaborator will be outside the comfort zone, and that the 2 of you can meet at an interface that's novel. Whether here, in this case, it's biophysics and developmental genetics, but we're both interested in cell biology questions. If you can find that interface, that's where the newest technologies and where the crosstalk between the 2 fields can really occur, and we can hopefully make great progress.
Oh, right on. That does make sense. We do see a lot of that push with NIH, standard funding that we all sort of chase. They love seeing a collaborative, highly integrative approach. And so that does sound like that is, the standard recipe moving forward.
Yeah, and students and faculty, students and labs, they shouldn't be afraid of this. And so if you want to move into a place where you're using C. elegans as a transgenic model, find a collaborator. And if you have a collaborator down the hall, great. But if you don't, there's companies like yours that, you can collaborate really well with you. And we've done a couple of collaborations with you guys now, where we're designing mutations into lamins, which are involved in tons of different diseases. And it's been a really fruitful collaboration, because you guys can crank the transgenics out faster than we can. And now we can use our skills to analyze it in our assays of nuclear movements and things like that. And it's just, there's no excuse to stay in your silo in today's science. There's no such thing as a cell biologist or a geneticist. These categories are all merging now, maybe under some greater molecular biology or something. But you have to be flexible and be able to move between things, move to the system that's easiest to study your question.
Yeah, I've heard that. I said before that even your standard animal models may not be the best, and there might be alternative animal models to use that are best for answering a very, a particular specific question. You mentioned again this one about the C. elegans being a system for looking at cell migration. Perhaps you could describe that one a little bit more. We have probably about a minute left, and would love to hear about how you think that is a great model for.
Yeah, so because C. elegans has this clear skin, you can actually watch nuclei moving around throughout different stages of development. And there's this one, we watch nuclear migration events, and in one event in the embryo, it's using a plus end-directed motor to move this way. And then another event in the larva, it's moving a minus end-directed motor to move the opposite direction of the microtubules. And we're trying to get at the interface right now. One of my students is trying to figure out how does the nucleus bias towards one way or the other way. And then we're also finding out there's other pathways, including an actin, a branched actin pathway that's pushing on the nucleus to help it squeeze through those narrow spaces. And we're also finding out that something about the heterochromatin and the stiffness or softness of the nucleus can help it migrate or not migrate. We're trying to work out the combinations, all three of those mechanisms to how the nucleus moves around.
Oh, right on. So that then you can imagine for a metastasizing cancer is it allows you to look at. I mean, how do you look at a metastasizing cancer in a human and be able to see that transparency that's not there? So that's the unique advantage, I guess.
Yeah, we have a genetic system. We can do 4 genetic screens to look at a normal process of development as the nucleus is squeezing through, whereas our imaging is not as good as a nucleus migrating through a microchannel or something. But we've got the genetic approaches that we can do.
All right. Well, it looks like we have about another 2 more minutes here, and perhaps the some of the other stuff we could talk about is after the Allen grant. What do you imagine you're going to be doing then? Let's look way down the road. Imagine trying to imagine what's going on after the Allen grant.
I would love to figure out a way to keep Gantz here so we can keep this collaboration going and keep the momentum going. And then we can just, after we start to learn more about these mechanical properties of the cell, then we can start to apply them to disease tissues or disease models to see how do the mechanics of inside the cell affect the pathogenesis of these diseases. Ultimately would be the long-term goal.
All right. Well, it looks like we're coming up on the end of our time. It's been a great conversation with you, Dan, and I appreciate you taking the time to speak with us.
Thank you, Chris. It was a lot of fun, and it's been a lot of fun working with you guys the last couple of years, and I highly recommend that.
All right. Well, I appreciate the plug, and it has been very, very fruitful. We have had some rare disease work with the UDN. You're talking about the LMNA work. We've got collaborations there. So, like you say, the collaborative landscape and developing the collaborations. We pride ourselves at In Vivo Bio for that, and it sounds like you've been chasing that and having great success. It allowed you to get the Allen grant, and that's a really amazing opportunity that you were able to land, and it sounds very exciting where you're going to be going. So thank you for speaking with us today.
Yeah, thanks for this opportunity to let me tell the whole listeners out there what we're up to.
All right. Thanks, Dan.